Hi everyone. So let's go ahead and take a look at section 6 of chapter 19. And this one we're going to be taking a look at the stimulation of the heart. Really what we're going to be taking a look at is diving into those little nodal cells of the conduction system. We're just going to look at a little bit more at their electrical activity, and then that's going to help us understand what's going to happen to the cardiac muscle once they go in and stimulate the cardiac muscle to actually contract. So let's go ahead and get started by, again, looking at these two types of cells that we are going to, or I guess group of cells that we're going to have in the heart. Remember that most of your heart are going to be these cardiac muscle cells that are going to have that contracting ability. That's where the muscle contraction comes from that is going to pump the blood through the heart. But remember that these guys cannot contract unless they get an electrical stimulus. And that's where your conduction system is going to come from. The little SA node, AV node, the bundles, the branches, all of those nice little structures we looked at last section. So they are going to have these things called nodal cells. They're going to be made up of these nodal cells, and these nodal cells are going to be these cells that are able to depolarize on their own, reach threshold on their own, and propagate an action potential as well. Exactly what this is going to need. So as you can understand, they're going to sit pretty much right next to the cells of the cardiac muscle system and help kind of sweep that action potential across the contractile cells. So that's what we're going to be taking a look at in the next section. So let's go ahead and look at this little guy a little bit closer here, these nodal cells. So they're not an actual neuron, but they are going to be these cells that I guess are neuron-like, right? If you think about how they are going to have these channels and things like that, resting membrane potential, but really that's the only thing that they have, and that's similar to neurons. Other than that, they are going to be completely different. But they're able to spontaneously depolarize and generate an action potential. So these are going to be the actual pacemaker cells that initiate the heartbeat. And of course, these are found in the SA node. We'll talk about how you are going to have them all throughout the conduction system. So let's take a look at what they have. They are still going to have very similar to what we've seen in 2401. In their membrane, they still have those potassium and sodium leak channels. They still have sodium-potassium pumps as well. Those are going to help set a resting membrane potential. And the resting membrane potential of a nodal cell is actually negative 60 millivolts. A little bit different than what you saw with neurons that are negative 70. Muscle cells are negative 90. These have a resting membrane potential of negative 60 millivolts. Let's take a look at some other interesting channels they are going to have inside of this plasma membrane. Other than the little leak channels and the sodium-potassium pumps, they are going to have some voltage-gated channels. They have sodium voltage-gated channels. They have calcium. Calcium's kind of a new one, right? And we're going to talk about its importance here in a second. And also potassium. So sodium, calcium, and potassium. So remember that the SA node cells, the nodal cells that are found in the SA node, exhibit something called autorhythmicity. This is the same as something we introduced before called automaticity. I introduced that term in another slide earlier in this in an earlier section. So remember, that just means that they are able to spontaneously fire. And I've said this lots of times before. Spontaneously fire, depolarize on their own, initiate the action potential, propagate the action potential. So that's what that means. This is going in and looking at the typical depolarization and repolarization. So, and it's going to be autorhythmic. It's going to have a nice little set pattern to it, and all of that good stuff right here. So let's take a look and see what's happening to be able to reach threshold, what happens at depolarization and repolarization. We're not going to go into all of the little nitty-gritty details about this channel's opening and this channel's opening. Yeah, it talks about it right here, and if you're interested in it, then read it over and know about it, especially if you're going into the cardiac field. It's really important to make sure that you know this information. But really what's happening here is that you are trying to reach threshold. So you're trying to get from that -60 to that -40. And it's typical of what we've seen before. It's going to be because of sodium channels, which is, it's typical. Let's take a look and see what happens once it reaches threshold. This is where it's a little bit different and it gets a little bit interesting. Actually, what causes depolarization in these nodal cells of the heart is calcium channels. Remember back whenever you looked at neurons and skeletal muscle, depolarization was caused from sodium channels? Here in the heart, it is going to be these calcium channels in the nodal cells. So that's kind of interesting. So remember, depolarization is going in and causing it to become that that membrane potential to be more positive. So it goes from that -40 to that 0. So that's basically all that's happening here because of calcium. And again, you don't have to worry about which way is going, which way to which way. Which way the calcium is flowing. Not all the little details like that. Just the interesting ones, right? You might not find them interesting, but I find them terribly interesting. All right, let's take a look at what happens here. Remember this one that is going down back to resting membrane potential? Remember, that's called repolarization. And that's what we are having happening here. And it's potassium again, doing its repolarization kind of thing. That's what it does. So it's just going to go through this whole process over and over again and just fire off over and over again. So the one at rest, 1 SA node's action potential starts at about 0.8 seconds after the last. So that translates to about 75 beats per minute. Inherently, the SA node would be a little bit faster. It would fire at about 100 per minute. But remember that we have that parasympathetic or I guess not stimulation, but the parasympathetic innervation that's going in via the vagus nerve and telling it to slow down. That's called vagal tone. Vagal tone is going to go in and set the heartbeat at a slower rate than what would normally fire off. So that's what we have here, this pacemaker potential, which is going to be how you have these pacemaker cells that is going to have the ability to reach its threshold without a stimulus. So let's go ahead and take a look at the pathway that this stimulus, this impulse, or this action potential, not stimulus, because it says right here it's without a stimulus. So let's go ahead and take a look at the pathway of that action potential or impulse. So now we're going to use what we learned in the last section, looking at the parts of the cardiac conduction system, and then we're going to see how the action potential spreads. All right, starts at the SA node, right? And then it's going to sweep across the atria. That action potential is going to sweep across the atria and go to the AV node. So when it sweeps across the atria, that action potential is telling the cardiac muscle cells to contract. And so that's exactly what is happening here, is those muscle cells are going to slowly contract. And I guess not slowly, this whole process takes 0.8 seconds, so it's not too slow. So it goes over to, eventually it reaches the AV node, and then that AV node, the signal is going to be delayed for a little bit, not too long at all, but it's going to be delayed enough to make sure that, since we just contracted the atria to move blood down into the ventricle, we need to make sure that we get all of the blood in there. So that little delay that you have here at the AV node is going to allow the ventricles to fill up. From here, we need to start going down and stimulating these ventricular walls now. So it's going to move through that interventricular septum. It's going to go down that bundle of His, and then from the bundle of His, remember, it splits into the left and right bundle branches. That way it can go and stimulate the left and the right ventricle. From there, it goes to the Purkinje fibers. Didn't like that at all. from there, it goes to the Purkinje fibers. Look, it goes up into those little papillary muscles as well. And we'll talk about what it does here in a second. So yeah, that action potential is going to spread through the gap junctions that we looked at in those intercalated discs, and that's what's going to allow it to kind of beat in that synchronous motion. So the contraction is going to start down here at the apex, and it's going to push blood that way. And that's exactly what we want. So the cells of the two ventricles are going to contract almost simultaneously, and that's exactly what we want happening. So that is how that action potential moves through the cardiac conduction system, and how it's going to cause the muscles to contract. So the Purkinje fibers, let's take a look at some of these special characteristics that are found in the ventricles. Purkinje fibers only in the ventricles, papillary muscles only in the ventricles. The Purkinje fibers are pretty large in diameter if you compare them to other cardiac fibers. And what that means is that helps kind of sweep that action potential a lot faster. And that's going to also make sure that those ventricles are going to contract at the same time. The papillary muscles, remember, they are going to be anchored to the cords that are going to be attached to the tricuspid and bicuspid valve. And so the papillary muscles are going to contract at the same time, pretty much almost immediately. And what they do is, remember, they are going to go in and pull on those cusps, and they are going to make sure that those valves are going to stay closed, because at this point, whenever the ventricles are contracting, or trying to move blood through the great vessel. So we don't want it to go back up in here. We want these guys to be closed. So that's why it's so important to make sure that they are closed. And of course, I already talked about how stimulation and of course contraction begins at the heart apex to make sure it ejects where it needs to eject to, right? So what's interesting about the whole system is if one kind of peters out, then you do have a backup mechanism that's not as efficient, but you have it, and it's better than having nothing. So you have the SA node, but you have something called ectopic pacemakers, meaning that you have pacemakers other than the SA node. So you have the other systems that are able to go in and depolarize on their own. I say system, but I mean the other nodes. That's what I was looking for. The other nodes that are able to go off and fire on their own, but they're going to fire off at a much slower rate. If you take a look at this, remember that the SA node typically is 80 to 100. I know the other slide said 75. But usually you kind of see this number 70 to 100. Once it gets down to the AV node does have an inherent rhythm. It can depolarize on its own. It's 40 to 50 beats per minute. So no, it's not very fast, but it's enough to at least sustain life. So this is obviously something that, if your SA node does stop working, at least you do have this kind of backup mechanism to make sure that it keeps on, your heart does keep on beating, but it's not going to be efficient enough to last for a very long time. And then if the AV node peters out, if it completely quits, there's no way that the other cardiac muscle, the other nodal cells found within the bundles and the bundle branches are going to sustain life. They fire off at 20 to 40 beats per minute. So that's why a lot of times if an individual's SA node stops working, they get one of those mechanical pacemakers, which is a device that is implanted, and it is going to go in and tell the SA node, hey, wake up, fire off. So it's going to make sure that that SA node is working properly. So what we're going to take a look at after this next section, we're now going to take a look at what's happening with the cardiac muscle cells. So this were those electrical cells, those nodal cells that were stimulating the cardiac muscle cells to contract. So now we're going to be taking a look at what happens within those cardiac muscle cells. So until next time.